Good morning, everyone. Good afternoon and good evening to everyone joining us, and welcome to today's webinar. Today's talk is entitled Assessing Activity in Early Phase Trials in Oncology, Current Designs for Expansion Cohorts in Phase 2. My name is Kelsey Brown, and I am the Director of Medical Writing Solutions at TransPerfect Life Sciences in London, and it's my absolute pleasure to be your moderator for today's session. So today's event will run for approximately 60 minutes, and we'll have some time towards the very end for a Q&A session with our speakers. So just some general housekeeping. This session is designed to be interactive, so it works best when you're involved. So please, throughout the course of the presentation, feel free to submit questions and comments for our speakers by using the questions chat box in your control panel. And we'll answer your questions towards the very end at the Q&A session. If you require assistance, reach out to me at any time by messaging me in the control panel that you have there. And at this time, everyone is in listen-only mode who's participating. Please note this session will be recorded and it will be made available to you on the IDDI website for viewing about 24 hours after the webinar is complete. This event is proudly hosted by the International Drug Development Institute, IDDI. IDDI is an expert center in biostatistical and integrated eClinical services for pharmaceutical and biotechnology companies in several disease areas, including oncology and ophthalmology. IDDI optimizes the clinical development of drugs, biologics, and devices thanks to proven statistical expertise and operational excellence. Founded in 1991, IDDI is headquartered near Brussels, Belgium, with a U.S. Center of Operations in Raleigh, North Carolina. Now, it is my pleasure to introduce you to today's speakers. We have Elizabeth Else-Court, Principal Statistician of Consulting Services at IDDI, and Dr. Everardo Saad, Medical Director at IDDI. So I'll let you two take it away from here.
Thank you, Kelsey. I hope everyone can hear me well. Hello, everyone. Thank you for attending our webinar. This is the last one this year. It's been a busy year for us, but we're already preparing our series of webinars for next year. And by the way, if you want to suggest topics, there'll be some space for that in the survey towards the end of the presentation. So this, for some of you who may not know, this webinar today is sort of a continuation of one Else and I did in October. So there we spoke mostly about phase 1 trials, and here we'll talk about expansion cohorts that may be part of phase 1 trials as well as phase 2 trials. And so you can find the previous webinar on our website if you haven't seen it. Now, today, Else will take the bulk of the presentation, and she'll tell us a lot about different types of designs that we think are useful for oncology. So she'll provide us with a taxonomy of the current, let's say, most frequent, commonly used frequentist and Bayesian designs. She'll talk about their key statistical properties, and importantly, points to consider when selecting among these designs. I will come, let's say, slightly more than halfway through to give Else some break to catch her breath, and I'll talk briefly about some approvals based on phase 2 trials. So now I will hand it over to Else.
Yes, thank you, Everardo. So like you said, this is in a way a continuation of the webinar we did in October. It's again an overview of different study designs, but now really focused on phase 2. And I have to say that it's slightly more technical than the previous one. But I would like to ask you, please bear with me if you're not a statistician. I just try to give as much theory as is needed, and I will always follow up with some examples. So don't be afraid when you start hearing a lot about type 1 error and type 2 error and some formulas. I will always try to give some examples to make it more concrete. So today's topic, the phase 2 studies, these are really learning studies. So these are studies in which we will collect all the information we need to decide whether our experimental treatment in this particular setting warrants further large-scale development. And so what we can do in a phase 2 is confirm the dose, identify, refine the patient population, and importantly, we will also characterize the drug efficacy and have a better idea or a first idea of the magnitude of the effect, which we will need to design a proper phase 3 or a pivotal trial. Now, those phase 2 trials, they should not take too long, and for this reason, they have a limited sample size, and the endpoints that we use, these are early efficacy endpoints that we think are sensitive to drug effect and are in a way necessary for an effect on the final endpoint. And the best known example, of course, of this typical phase 2 endpoint in oncology is objective response rate that is based on tumor response, and that is used as endpoint instead of the final long-term endpoints PFS and OS. We have to keep in mind, though, that those early endpoints are more often than not, they are not perfect surrogates of these long-term endpoints. And so if we see an important endpoint on this, an important treatment effect on the early endpoint, this does not necessarily will translate into an important treatment effect on the long-term endpoint. Talking about the taxonomy of phase 2 designs, I think they can largely be classified according to those lines. So we have mostly single arm designs, sometimes randomized trials. From a statistical approach, we have Bayesian or frequentist designs, and we can have designs with or without interim. And let me first talk about the basic designs, the single arm, and the randomized design. In a single arm design, you will enroll patients, and all of those patients will be treated with your experimental drug. You will follow up the patients, and you will compare the outcome of those patients with an historical control. So here we know that selection bias is always possible. And we know that potentially this historical control that we are using, this might be outcomes that were obtained under another standard of care, so that by now the standard of care might be better. And so this makes that no historical control is perfect, right? This historical control can just be a treatment outcome, a response rate that you find in literature, or it could be also individual patient data. In a randomized comparative trial, you randomize the patients that are enrolled, and you will actually treat patients with the experimental drug and with a placebo or standard of care therapy. You will follow up the patients, and the main analysis of this trial will be a comparison between arms. So here, in fact, the trial is designed as a phase 3, but often with these earlier endpoints and with a larger target effect size and more liberal error rates to keep the sample size at a phase 2 level, say. But most of the trials in phase 2 will be single arm trials, and I will also come back to some intermediate solution. In terms of the statistical approaches, I think the most known approach and the most commonly used in clinical trials is the frequentist approach, frequentist designs, and I will try to explain based on a single arm trial. So we will test in a single arm trial one experimental treatment. We will observe the response rate and compare it to a historical control rate, say that is 20%. And in the frequentist world, we will define the null hypothesis here, saying that our response rate or our parameter of interest is below 20%, and test it against an alternative, which is that the response rate is at least 40%. We will reject this null hypothesis, meaning that we will decide that our experimental treatment is better than this historical control based on the probability of the data, given that the null hypothesis is true. And let me explain this in a bit more detail. So it means that we will think about the true response rate of the treatment as being fixed. And we then think about repeated testing. So if we would do the same trial many different times, what would be the probability that we observe the data as we have observed them, or even better, if this null hypothesis is true? And if this probability becomes too low, so below 5%, for instance, below our alpha level, then we say this null hypothesis cannot be true. Now, this repeated testing principle is also the most criticized point, I think, of the frequentist statistics in general and designs, because of course you are not going to do many of those trials. It's just a theoretical concept. It's a thinking experiment, and it's a way to decide whether you can reject the null hypothesis or not. So the true response rate is something fixed, and our data are random, because if I would do the same experiment again tomorrow, I would end up with other data. So each data set is a kind of random sample of the same population. If we want to design the trial, what we need is clinical input. We need to know what is our uninteresting response rate or our historical control, what we want to beat. And we also need an estimate, a guess of how good our experimental treatment could be, what is the response rate that we expect. And it's really these two response rates that drive the sample size. The larger the difference between both, the smaller the sample size, because to pick up a huge effect, you just need a few patients and the other way around. Apart from this clinical input, you also need to fix the operating characteristics, our type 1 and type 2 error. Type 1 error are false positives, right? And here, the typical frequentist interpretation. So if this null hypothesis is true, what would then be the probability to observe our data and to reject this null hypothesis? We typically set this for a phase 2 at least, we set this at a one-sided level of 5% or even 10%. And our type 2 error, or our power, here it's our false negative. So if the true response rate is really above our historical control, then we really want to reject it, right? We want a high probability of finding it. And this power is often set for a phase 2 trial to 80%, whereas for later trials, it often goes above. But depending on the situation, it's your call to set it where you think it is best. What I wanted to point out is that in a single-arm trial, rejecting the null hypothesis is sometimes interpreted in a slightly wrong way. So the correct way of interpreting the rejection of the null hypothesis means that you have shown that your response rate is above this historical control rate that you want to beat. And that your data, they are not inconsistent with the response rate that you used as alternative hypothesis to design the trial. But in many scenarios, when you reject the null hypothesis, you have observed a response rate that is well below this alternative response rate. So it does not mean that we accept it. It does show that it's really important where you set the bar for this low response rate for your null hypothesis. If you set it close to, for instance, the response rate that you can achieve under the standard of care, then if you can reject it, it means something. And it means there's something interesting in this trial, and you could think about further development of the compound. If you set this bar too low, below the current standard of care, well, you haven't shown anything of interest, because nobody is interested that you are better than some very, very low response rate that even the standard of care can do better. So once we have all these design parameters, we have to define the sample size and also the threshold for the number of responders to take our decision at the end of the trial whether we think the trial is successful or not. And in a frequentist design, let me guide you through an example to show you how we could come to this sample size. So say we have a sample size of 15 patients, and we have a null hypothesis of a response rate of 30%, and we would like to test it against an alternative of 60%. So first, let's look at the null hypothesis. It says that the response rate is 30%, and this means that if we would do an experiment many times with a true response rate of 30%, here you see what the probability is we would have all these number of responses. So we would have here about 20% of trials that would come out with 5 out of 15 responders in this trial. If we would set our threshold at 8, it would mean that if this null hypothesis is true, only a very small number of trials would have a number of responses that is above this threshold. And this small proportion is below 5%, so it means we can use the threshold of 8 responders and safeguard or control our type 1 error. Fine. Now we look at the power, the type 2 error, and we will change our mindset, and we will now assume that the alternative hypothesis is true. And we assume that the response rate is 60%. But if we now look here, what is the probability to have a trial with 8 responses, assuming that the trial has the treatment has a response rate of 60%? You see that it's still somewhere in the middle, and it's not enough to have a power of 80%. To have a power of 80%, we should set a threshold here at 5 responders, and then we have 80% of probability to have a response that's above that. So it means that with a sample size of 15, we cannot find a good threshold that both satisfies our type 1 error and our type 2 error constraint, right? So it means we need more patients. So suppose we now increase the sample size from 15 to 17, looking at 8 responses on 17 for the null hypothesis. This still looks good. It means we only have a very low probability that we have more responses if the null hypothesis is true. And at the same time, if we now look at the alternative hypothesis where the true response rate is 60%, we see that this threshold of 8 responders is a good one because we have 80% probability to have a higher number of responses. And it means that with a sample size of 17, we can find a threshold that is both satisfying our type 1 and our type 2 error that we would like our design to have. And that's it for the, let's say, more theoretical part on the frequentist designs. And let me now show you some simple examples. So one of the designs, the simplest design, but still often used, it's the Fleming one-stage design. Very simple. You set your design parameters, your alpha, your beta, and your 2 response rates corresponding to your hypothesis. You calculate there is only one unique solution in this case for a minimal sample size. You enroll this number of patients, and in the end, you decide based on a predefined threshold. So, for instance, if you have an alpha level of one side at 10%, a beta of 20%, and you are comparing response rates of 40 versus 60%, the Fleming one-stage design would result in 30 patients and your threshold in 15. It means you enroll 30 patients. If you have 15 or less responses, you say that your trial is futile or your experimental treatment is futile in this setting. If you have more than 15 responses, it means that you have rejected the null hypothesis and you can think about further development of your trial. I know this is an oversimplification because in a phase 2 trial, I don't think that any sensible person would only look at the primary analysis and the primary endpoint. But I mean, in terms of the primary endpoint, it would mean that you have succeeded. And what I also wanted to point out is that here, when you have the minimal number of responses to be successful, meaning 16 out of 30, you have a response rate of 53%. So that's, as I said, it's below the 60%. So you have not shown that your response rate is 60%. You just have shown that you are above 40%. and above 40%, you can see here, when you construct an exact 80% confidence interval, you can see that this 40% is just outside of your interval. And I use here an 80% confidence interval. That is because we have a one-sided alpha of 10% corresponding to a two-sided of 20%, right? So saying that you rejected the null hypothesis is equivalent to saying that your P0, so your historical control rate, your low response rate, is not contained in the confidence interval for the observed response rate. You can make it slightly more complex by including an interim analysis, and that brings us to the also very well-known Simon two-stage design. In this case, you define your design parameters, but there are different possibilities to optimize it. And so maybe first, the Simon two-stage design, it means that you have first you enroll a lower number of patients. After you have observed the endpoint on these patients, you will decide whether the study is either futile if you fall below a threshold, or continue otherwise. If you continue, you add other patients, and in the end, you again compare the number of responders with the threshold. Now, in terms of defining the sample size, it could be either that you minimize the expected sample size under the null hypothesis. If you use this optimality criterion, you call it an optimal design. If you say that you want to minimize the total sample size, so the maximum sample size, it's a minimax design. But there are still many other designs that we call admissible in between the optimal and the minimax that all have good properties, and that you could also select if you think these suit you better. They differ in the timing of the interim analysis. So if one design has an interim after 10 patients, then another will have an interim after 12 patients. And potentially, if you want to fit this phase 2 in a design, in a study together with the dose finding design, and you know you will have, say, 12 patients treated at the final dose, you could select an interim after 12 patients, just as a practical example. So in practice, how does this Simon two-stage design looks like? If we say, again, we use the same assumptions as the same design parameters as we used for the Fleming's design, we can, if we say we take a minimax design, so we want the design that minimizes our maximum sample size, that would then result in a design of a total of 28 patients. But here, because it's a two-stage, you first enroll a smaller number of patients, 16 patients. You check how many responses you have on this number of patients, and only if you have more than 6 responses on 16 patients, you will continue to the stage 2. You add 12 more patients, so for a total of 28, and then you see whether you have more than 14 responses on 28 or not. In case you take the optimal design, you will end up with 38 patients, right? And you will have different sample sizes in the first and the second stage, and somewhat different thresholds. Here you see again that when you are just, you have the minimal number of responses to have a significant trial. So in this case, it would be 15. You have observed 53.6% as your response rate, and your confidence interval also excludes your low response rate that you wanted to beat. But here, this is not just a simple exact confidence interval, but it's a type of confidence interval published by Koyama that takes into account, say, the adaptiveness or the two-stage feature of the trial. There's also, as an example, a Fleming two-stage design that will then add on top of the interim for futility and interim for efficacy. And then you have, again, more optimality possibilities. I must say that not many of our clients are interested in seeing this efficacy analysis at interim, because most of the time, if you have a treatment that is active, then you will continue development and you want to collect more data, you want to treat more patients to have more information to do to prepare for the design for your phase 3 or later phase trials. One thing that I wanted to mention about this type 1 and type 2 errors is that there is a design and there's an attained or a reached error level. So if we say that we want an alpha level of 10% and a beta level of 20%, it means that in the design we will constrain the design to have error levels that are below these design settings. So in this Simon two-stage design that I just showed you with 38 patients, the attained alpha was 9.8%, so it's not exactly 10, and also the type 2 error is not exactly 20%, but that's because of the discreteness of the binomial distribution. You will often, or you almost never have the exact design settings. Now, if you still constrain those type 1 and type 2 error, but you allow for a small tolerance, you can end up with with a different design and a different sample size. If we say, well, we want alpha to be constrained at 10%, but we can live with a 0.5% difference, then this design would satisfy that. And you would have a lower sample size, but you would have an alpha that's somewhat higher. And why I'm saying this is because if you use different softwares or you read some sample sizes and you see different values, often what is different between the different numbers is really this tolerance level, right? So it's just something you need to be aware of. Now, those frequentist designs, they are very simple to use, but of course, it's very strict in terms of the sample size that you need to evaluate your experimental treatment, and also in the stopping boundary that's defined only for this specific sample size. And we know that in reality, the actual sample size can often be different from the planned one, especially if you have a multi-center trial. And then we try to be pragmatic and say, okay, if we have an interim analysis at 20 patients and we have observed 22, well, we take the first 20 for the formal interim analysis, and then we continue. But of course, this is not optimal. Another thing about interim analysis is that will the sponsor stop the accrual while you are following up the patients you need for the interim? For instance, if the response rate is evaluated up till 3 months after treatment, while you are following up the patients for the interim analysis for 3 months, if you don't stop the accrual, potentially you already have enrolled all the patients that you need for the second stage, in which case that the interim loses a lot of its interest. Also, I think we always have to warn for the sharing of information, sharing of study results for a trial that is still ongoing, as this can open, of course, the door to operational bias and jeopardize the validity of the trial. We know that investigators and everybody involved in the trial, they are all subjective human beings, and consciously or unconsciously, they can, for instance, enroll other types of patients once that some summarized interim data become available. I now change gears and I look at the other statistical approach, the Bayesian approach, and it's a different concept. It's a different way of looking at data and probability. So if we think again about this single-arm design in which we want to compare our response rate to an historical response rate of 20%, and we hope to do better than that. Now, before the trial starts, in the Bayesian world, we have a prior belief about our parameter of interest. So here our response rate. And we will then observe the data. And if we think that this prior belief is correct, what is then the likelihood of observing this data that we have observed? And we will combine the prior, we will update the prior with the data we observed to obtain a posterior belief or a posterior probability that is based on having seen the data and the prior. So you can see here that the data are considered as fixed. So we have observed them, they will not change anymore, but it is the response rate that is unknown and random. So here the probability, it's no more the probability to observe the data if the null hypothesis is true, but it's really the probability that is quantifying our uncertainty on this response rate. And what is depicted here on the graph is that often in a phase 2, we don't have a lot of prior information on our response rate. So we use what we call an uninformative or a flat prior, meaning that we think that every response rate is as likely to occur. We know nothing. But once we have observed the data, we think that our posterior probability, we think that our response rate is around 30%, but still with a lot of uncertainty. So yeah, like I said here, it's really, we are looking, the more data we have, the more we know about the true response rate, and the lower our uncertainty becomes around this response rate. There's no longer this frequency in the long run. There's no true responder rate, and for many people, this feels like closer to the actual clinical decision process. What I've shown on the previous slide, and here it's Bayes' theorem, showing that we will combine data and prior. And this prior, in fact, you can see it in an loosely speaking way as adding data. If you use in a non-informative prior, you let the data speak for themselves. You can use fictitious data, so you can ask an expert, what do you think? And add this as data. Or you can use real data from prior studies. For a Bayesian design, your sample size calculation will be defined or will be done based on a threshold for posterior probability that you define. So in order to reject the null hypothesis, how large should your probability be? The probability that after seeing the data, that your true response rate is above this historical control. And for instance, let's say we want it to be 90%. And then this design will be simulated under different scenarios. We will also look at this design simulated under the null hypothesis that will give us the type 1 error, and we will look at it under the alternative, so it will give us the power. And you see here that those concepts of type 1 error, that's a clear frequentist concept, but it's often, I don't think that many Bayesian designs are used in a clinical trial for which the type 1 error was not checked. When you read about designs using Bayesian probabilities, you will come across posterior and predictive Bayesian probability. And as a rule of thumb, when we use data for a go, no-go decision, a Bayesian posterior probability is mostly used when you ask questions of the nature of, is there convincing evidence in favor of the null or the alternative? And this is something that you, in a frequentist design, would answer with a p-value or an estimate. Whereas if you have a question of the type, will the trial likely show convincing evidence in favor of the alternative if we add the data, then you look at, you will be better off addressing this with a Bayesian predictive probability. In a frequentist design, this would be a more conditional power. And I will briefly go over some Bayesian designs, and you will see that this is closer to the decision-making process. So a Bayesian interim monitoring with predictive probability, it starts from this predictive probability concept, which is the probability of rejecting the null at the end of the study if the current trend continues. So in English, it means, given the current data, what is our chance of a positive result at the end of the study? And I think this is the type of questions that trialists will relate to and will want to answer. In case this probability of being successful in the end is already very high or very low at interim, in fact, we can say the answer is as good as known, and we can stop the trial and draw a conclusion. And these predictive probability can be easily calculated after observing the response for each additional patient, in which case you have a continuous monitoring, or you could define, say, a cohort of 5 patients and say, and that you will look at this predictive probability after seeing the results of 5 additional patients. When I say easily calculated, and I won't go through this, it just showing for the statisticians that it only involves calculating some beta and some beta binomial distributions. And of course, we have to make a decision on what will we call very high and what will we call very low, what will be really the boundaries or the threshold to stop the trial for futility or efficacy. Now, again, for efficacy, usually or in many cases, this boundary is set to 1. So it means that you should have 100% success guarantee before you stop for efficacy, meaning you will not do that. If you don't stop for futility, then you continue. And in the end, you will stop if your posterior probability is above the threshold that you defined. If not, you will accept the null hypothesis. Now, to select all these boundaries and the maximum sample size, controlling a type 1 and type 2 error, this is something that is done by a trial and error approach. It's a simulation-based approach, and the Simon's two-stage design is often a good starting point for that. So you see that here it's a Bayesian framework that we use as a tool to design clinical trials that still has desirable frequentist properties, right? And another way I think in which this type of design is often used is for a continuous monitoring for toxicity. For instance, you just have treated a couple of patients in your previous, in your phase 1 trial, just a couple of patients treated at this dose that you want to carry forward. Then you add some continuous monitoring of the toxicity so that if you increase a certain level, that at least there's a trigger to have a safety monitoring committee or to have a detailed look at the data. And just to give you an example, so for instance, here above you see the Simon two-stage design that we discussed, and below is for the same design parameters. I'll have now a predictive probability design. So with a maximum sample size of 28, when I say I want to have an interim after 5 patients have been treated and evaluated, but I only want to start when we have at least 10 patients, because if you have really small numbers in the beginning, I mean, let's say the confidence you can have in the response rates that you observe on 2 or 3 patients are really very low. And so often the first interim monitoring is started a bit later. And if we then set some boundaries, we have the design that is shown here in the table. So you start monitoring at 10 patients, and if you have 3 or less patients, you stop the trial for futility. You look again at 15 patients. There the threshold, the upper limit of the threshold is 6, and so forth. If you don't stop for futility, you continue until you have 28 patients, and then you will consider the drug efficacious if you have at least 14 responders. So you see it's very close to the numbers of the Simon two-stage design, but here with those settings, I have an alpha level that is above 10%. So if I don't like this and I wanted to bring this down, I should probably increase my sample size somewhat. The last design I would like to discuss is the Bayesian optimal design for phase 2, and throwing in another acronym, the BOP2. And this is really cool because it explicitly controls the type 1 error rate. So it's kind of bridging the frequentist and Bayesian designs. And it's optimized for a given sample size, maximum sample size, and a type 1 error. There are 2 optimality criteria. And again, the stopping boundaries can be pre-specified in the protocol, which is it's very easy and it's very clear to everyone what you will decide when. And the cool thing is that you can also handle more complex endpoints and objectives. And let me just show you one more example. So you could look at a simple binary endpoint like we have done in the other trials. But let me also talk about this fourth example. So for instance, if you say, well, I think this trial is this drug is interesting if I have a response rate that is at least 45%, but at the same time the toxicity rate should be below 30%. Because if either of those is not satisfied, then I'm not interested in this treatment to develop it any further. So this is also, let's say, a multiple endpoint or a multiple objective that we can deal with in this type of design. And another nice feature of this design is that the boundary adapts to the accumulated sample size. So it means that if you have more data, you will have more stringent boundaries. And it was shown that this substantially improves the power of the design, but it comes at the cost of a different clinical interpretation. And let me show you here. So for the example in which we have just the response rate that we want to test 20 versus 40%. After 10 patients, our threshold for futility would be 1, so that's a boundary of 10%, whereas after 40 patients, our threshold is 10 out of 40, so it's 25%. So you see it changes with the accumulating data. Now, and here the example number 4, where we said that we only wanted a treatment that is sufficiently active and is not too toxic. So it means that if you are either futile for your efficacy or you are overly toxic, you stop the trial. And so you have pre-specified boundaries for each endpoint. One thing to say still is that most of those Bayesian trials, phase 1 and phase 2, they were developed by or in collaboration with the statisticians at the MD Anderson Cancer Center, and they also have a really nice software website. It's the trialdesign.org, and you see they have the simple Simon twostage, but they have the Bob2, they have the predictive probability, and it's really user-friendly software. And yeah, if you're interested in this type of designs, I think it's a very good point to start exploring. So now, Everardo, after going over the different designs, I would like to hand it over back to you.
Yes, thank you, Els. Don't go away because it's going to go back to you pretty soon. So basically, Els, until now, she spoke about single-arm designs, and I just wanted to bring some, let's say, historical perspective to the value of these studies for drug approval. So we know that the landscape is changing in drug approval. We've seen this many times in different contexts, and basically what used to be a clearly demarcated transition between phases 1, 2, and 3, with activity being part of phase 2 and 3. Now activity has moved even to phase 1. And we know there has been a lot of regulatory activity recently in terms of providing guidance on how to do that. So among the many possible guidance documents, this one on expansion cohorts from about 2 or 3 years ago is a relevant one. And also we know that, for example, even some approvals have been based on not only one single study, but rather patients from a collection of studies, in this case, the approval of pembrolizumab for microsatellite instability high tumors, different types of tumors assessed in 5 different phase 2 trials. So it's more like a cohort type of assessment. And so this is clearly evolving, and so single arm evidence is accepted as sufficient for approval, and this is not new. Just to give one more recent example, not too long ago, this drug was approved for non-small cell lung cancer with this particular point mutation in KRAS, and obviously without randomization, the approval is based on anti-tumor activity as indicated by good response rates and also duration of response. This is beginning to become more relevant, not only in immunotherapy, but in this case with a targeted therapy. And we know this is not new. The FDA has said for quite some time now, and they periodically update the publications. This is already an old one, but we can see how many drugs are approved based on single arm or non-randomized evidence. So for example, in this compilation some years ago, of 53 approvals, 16 were based on data from single arm studies with no comparison group other than historical, like Els mentioned. And here I'm not making a distinction between accelerated or conventional. Many of these approvals are indeed accelerated. And also EMA does the same. So recently there's been some review papers listing approvals. This is not only for oncology, for other areas as well, but the list is very long. I just go all the way to C here, but it really goes all the way to Z for zinc. And so many drugs in different indications, not only oncology, have been approved on the basis of single arm studies, both by EMA and FDA. And finally, this also recent publication in which drugs have been approved based either on expansion cohorts or phase 2 trials in oncology. And so on the left-hand side, it's EMA. Most of the applications have been accepted, and for FDA, nearly 100% of the specific applications were accepted between 2005 and 2017. So that is just to say that approvals based on single arm trials are not unheard of. They are possible. Most will be accelerated approval or one of the fast track modes pending confirmation in a conventional comparative study. Now, before I hand it back to Els, this is more or less the discussion we have often with our clients. So basically, if you're planning to design a phase 2 study, you're faced with two obvious possibilities. Either here it's specifically thinking about an expansion cohort, but it could be applied to phase 2 trials. So you finish your dose escalation phase, and suppose you now know what dose to take further in development. So you can, for example, have different expansion cohorts depending on indications, having the null hypothesis coming from historical data, and then design your study seeking to find a certain alternative response rate. Or you can think about randomizing with against some kind of control. Control we may discuss forever, but it's not the point here. But this assumes that there is a standard control that can be used in a randomized setting. Now, the dilemma here is you're always the same. So you can do probably much smaller study by not randomizing. So this is more efficient. You will end earlier and spending less money, but at the cost of not having more information that you could have by having an internal control group rather than a historical control group. So by doing that, you would have a better estimate of the treatment effect that you expect and that you need, like Els mentioned, in order to design your phase 3 trial. So Els, let me hand it back to you.
Yeah, so let me come back to those two options that you laid out, Everardo. So you have the option of the single arm trial, the randomized trial, and let me give you an intermediate option as well. So just when this is, I think, a typical example of a sponsor that comes to IDDI to design a trial and says that they have a drug that if they would add it to a checkpoint inhibitor in lung cancer patients, PD-L1 positives, that they think it could increase the response rate. Say the response rate with the CPI only would be 40%, and they think they could increase it, and that would be clinically meaningful, to a response rate of 60% when they add their experimental drug. So we've discussed this, how Simon's two-stage design would look like. It would need a totality of, for instance, 28 patients that you enroll in 2 stages, and you enroll only the patients in stage 2 if you have passed a first futility bar. Only 28 patients, so very low sample size. If you would look at this comparison of response rate using a randomized comparative design, using the same response rates, then you would need 116 patients that you would randomize to experimental and control. So it's a huge difference, right? It's changing from 28 to 116. It does come with all the advantages of randomized trial. It has a potential interim analysis after you have included half of the patients, so that also you don't need to go beyond 58 patients before passing a futility bar, but still it's a higher sample size. Now, what is an intermediate solution that we sometimes propose is that we designed a trial as a single arm trial, for instance, with using the Simon two-stage design, but then we also add a calibration arm. And let me explain. So really the sample size is driven by a single arm design. It was 28 patients, and this is exactly the same as for a Simon two-stage. But what we do is that we add, for instance, 2-to-1 randomization so that we have a limited number of control patients. So if you stop at interim, you expect to have about 6 control patients. If you continue after the interim analysis, you have 8 more control patients. So you would end up with 42 patients, 28 in the experimental arm and 14 in the control arm. Now, it is a bit of an in-between because it's randomized, but it's not comparative, and your primary analysis will still be that you will still be only based on your experimental arm, and you will just look at your response rate in the experimental arm and compare it to the historical control. But you can do more. So you can have different outcomes in your experimental arm. Everybody wants to have outstanding results, but this is rather rare. So usually results are rather disappointing or they are promising. And I would like to show you how these internal calibration arm can be of use in the different scenarios. So if you would have only this experimental arm and you design the trial for comparing 40% versus 60% of response rate, this would be your outcome. You would say, well, it's promising because I have 60%, that's above 40%. It's disappointing, only 40, or it's really outstanding, I have 80%. And if you don't have this internal calibrator, this is what you have. And with this information, you should design your further trials. Now, if you also have a control arm, you can put the results observed in the experimental arm a bit in perspective. It's not meant to be formally compared, but for instance, if you have promising results and the controls are as you expected to, then you can be more confident to take this drug forward. If you see that also the control group has better results than expected, well, then potentially there's not a true treatment effect, or the effect that you see between the arms is not as large as you would anticipate by just comparing with your historical control rate, and you might want to think twice or look at other evidence before you continue. When your result is disappointing and the control arm, the observed results in the control arm are as expected or better, indeed, you're disappointed and it's best to stop the development. If also the control arm is worse than expected, then potentially there is some signal in here and it's worth further investigating. Now, most sponsors say, well, but we think we will have outstanding results. And in that case, this control arm will serve no purpose. And I think this is not true. I think mainly in this case, it's really useful to have this control arm, because if you have these outstanding results and you do have a control arm that is only as expected or worse, you see this huge difference, and potentially even with this small sample size, you can do some formal testing. If, of course, the control arm does better than expected, you might be a bit tempered in your enthusiasm and might think twice before going forward. So before, I think I have to end soon to have some minutes for questions. I wanted to show that what I've been discussing now, it's really the basic phase 2 designs, but these are underlying many of the other phase 2 designs. So we have, of course, basket trials in which patients from different histologies are treated with the same treatment because their tumor biology is similar. We have the umbrella trials in which patients, depending on their biomarker information, will be treated with different treatments. And these trials, these basket and umbrella trials, they can be randomized or not. They can be designed as independent trials, or they can be designed with borrowing information across the cohorts. All these things are possible. And then, of course, last but not least, we have many seamless phase 2, phase 3 trials. Seamless can be in two ways. So operationally seamless, meaning that you have within one master protocol, you have independent phase 2 and phase 3 study stages, and the phase 3 study is analyzed based on the patients enrolled in the phase 3 only. In this case, you can use the designs that we've just discussed. If it's an inferentially seamless design, then of course it's a completely different matter. If you for your phase 3 you want to include your phase 2 data in the analysis, you need more complex methods to control your type 1 error. And I think that would be that would be more than sufficient issues to discuss in another webinar. So just some concluding remarks. Phase 2 is really a learning phase in the clinical development. Bayesian framework, it provides a good platform for this learning phase, but we should not think about the study design to be either only frequentist or Bayesian. It can be a combination of both, and we should just pick the design that fits the situation best. And even if you would go for a single arm design, I think this internal calibration arm can be really interesting to give you more information about the activity of your drug of interest. And that was it from my side.
Well, thanks, Els. Will you switch on your camera too?
Yes.
So while people perhaps send in their questions, I have some already from the audience. So this person is asking you, Els, probably more than me, the following: if there is already validation of the MD Anderson software, if it's let's say a validation from the point of view of regulatory authorities accepting it, and also if it can be used to compute sample sizes, and if it can be used for trial design.
Yes, I think it can certainly be used. And if you look at their website, they say they try to validate it in many different ways. So there's a check by others. So there's one developer and one does the QC. They try to compare it with other software, but I don't think that it comes with any guarantee, right? But I think overall it's really reliable. I have never seen anything weird about it. But before I would put a final trial in a protocol, I think I would prefer to check it with another software like in R or something, just to be on the safe side. But I think in general, it's really reliable.
All right. There's another quick question about regulatory issues. In your opinion, are regulatory agencies open to this intermediate type of trial that you described?
Yes, they are now. So some years ago, when we first proposed this, they were, I think, a bit confused because it was like, what is it? Is it a single arm trial or is it randomized? So I think it's very important for this intermediate solution that you make it crystal clear what your objective is. But I think by now, yeah, I think we have designed several of those trials, and they, both FDA and some European agencies, they approved it. And I think in many situations, they see the added value, yeah, of having this internal control rather than just relying on this historical estimate.
Okay, I know we're at the top of the hour, but maybe you can
I talked too
address
long.
it quickly. No problem. No, it was great. Would the type 1 error be not be inflated for a Bayesian design with many interims or continuous monitoring?
Yeah. That will be a question, I think, related to the predictive probability design. So, well, if you simulate the properties of this design, so you have planned interims and so forth, and then your design should control the type 1 error for your planned interims and your planned thresholds. But of course, if you in reality end up with another sample size and you do something different, then that could affect it. It's robust, but in that case, I think it's safer to design the trial with just a somewhat lower type 1 error.
Okay, I promise it's the last one. Often efficacy data are collected in dose escalation phase already. Can we use this information in the expansion cohort, and how? Do we have to adjust for multiple testing?
No, I think then that's really at the sweet spot of the Bayesian designs, right? That you can use it as prior information. So that's one idea. For
I
your
think the question was more general, regardless of the statistical
yes. So you
framework.
mean that if you have like a phase 1 design and you have a phase 2 afterwards, yes, then you include the patients from your phase 1 design. You take them as participants for your phase 2, because I think in this case we are not talking about pivotal evidence. We are talking about proof of concept, and
okay.
so that should be fine.
All right, that's the reason why the potential inflation of type 1 error is acceptable, let's say.
Yes, indeed.
Okay, all right. Well, we have more questions. For those of you who have sent questions, we'll attend to them by email. And if yes, so I think we have to stop. We're 2 minutes past the hour, and so I'll hand it back to Kelsey. Thanks a lot, Els.
Thank you.
Thank you both. Thank you so much for your presentation. And thank you everyone in the audience for attending as well. If you submitted a question and we didn't get to it, we'll chase up with you after the webinar today. And if you have any further questions, you can just submit those to info@iddi.com. That's info@iddi.com. We will be sending out a follow-up email with access to the recorded archive of this event. So you do have access to that later on. And just a final thank you to our speakers, Els Kort and Dr. Everardo Saab. We really appreciate your presentation today, as always. And thank you everyone who's attending. We wish you a really great day, and thank you. Thanks so much.
Thank you. Bye, everyone.
Bye-bye.
Bye-bye.